تمام دكتور أمير, go ahead.
Thank you to everyone all of you for being here with us today. We are very pleased to be able to welcome you all to our new talk of the Molecular Genetics and Biotechnology seminars. Before I get started, I would like to express my sincere appreciation to Dr. Ahmed Salam and for all the organizers for their great efforts. During the next 40 minutes, we will have a talk titled The Bioanalytical Resources, Data Visualization and Analytical Tools for Multiple Levels of Plant Biology. This talk will be given by Dr. Mohammed Ali. Dr. Mohammed Ali is an assistant professor of crop genetic and breeding at Desert Research Center in Egypt. He received his PhD in 2018 from China. Dr. Mohammed has been working in plant biotechnology in the field of plant functional genomics and secondary metabolism for 14 years. His current research involves in the usage of different plant metabolites database and the new techniques such as RNA sequencing as a method for identifying and discover genes involved in the biosciences of various primary and secondary metabolite pathways in model and non-model plants. Dr. Mohammed has published many peer-reviewed papers in many different journals. I don't want to waste your time more than this, and please welcome Dr. Mohammed, and you have like 40 minutes for your presentation, and after your presentation we will have like 10 minutes for discussion. Please go ahead, Dr. Mohammed, and thank you very much again.
Thank you, Dr. Amir, for this introduction. First of all, it is my pleasure to be here today with all of you. Secondly, I would like to thank Dr. Ahmed Salam and his team members for this kind of invitation, especially Dr. Amir Effat and Dr. Abdullah Talat. My name is Mohammed Ali, and I am a researcher at Genetic Research Department at Desert Research Center. Title of our presentation or lecture today, the Bioanalytic Resource Data Visualization and Analytic Tools for Multiply Level of Plant Biology. Let us start with a brief introduction about this database. The Bioanalytic Resource for Plant Biology, or BAR, is a board for accessing large data set from approximately more than 15 different plant species, with focus on transcriptome, protein-protein interaction, and the promoter data. It consists of numerous databases for which it is currently have added useful metadata data visualization tool to display the key result from this database, and the visualized analytic tools to identify gene expression pattern of interesting based on public available data. In our literature today, we will give brief cover for some of these tools and scenarios in which they may be useful for plant researchers. The BAR database have approximately 145.2 million gene expression measurement. Display this data by using AFP or electrofluorescent spectrograph for exploring and analyze a large-scale biological data set. And as you see here, you can use this link for accessing to this database. This is database already developed and was developed and created by a researcher at the Department of Cell and System Biology at University of Toronto in Canada. This is, as you see here, this is a form of this database. As you can use, this database can be divided into three main sections. The first section is related about gene expression and the protein tool. The second is related about molecular markers and the mapping tool. The third is related about other genomic tools and widgets. Also, as you see here, each tools in BAR database represented by a small window, and this small window have three icons: info and go and the BAP. Info is abbreviation of information that is all researcher or created or developer of this database use this information from other research to build or to create this database. Go icons if you need to apply or need to use this database. Public abbreviation of publication if you need to you will find all the publications that use this database for some research or get information from this database or tools for research. Okay. Also, this database have tools for monocotyledon plant, okay, such as maize, rice, barley, and wheat. Also, This is database have tools for dicotyledon plant such as medicago, soybean, potato, tomato, and grapes, and stevia, and kiwi. This database also have other gene expression and protein tools like mice and humans. And this is also have tools that allow us to make Arabidopsis interaction viewer and rice interaction viewer. Also, this is database have molecular markers and the mapping tools. This is can use the 4+ digester and marker tracking and record generation mapping. As you see here, application of VAR. The data in VAR database can use can use by researcher to get information. This is will help us to study electrophoresensitive spectrograph development in our plant, and they can give us information about by expression angular. And this is data can be help us to predict our gene expression under biotic and abiotic stress, and also can help us to get information about or to predict information related with our gene expression in which organ in the cell, and also can be help us to find the interaction between our gene between our target gene and the other gene in the same plant. Here, as you see here, and this is window, this is the first tool for cell electrophoresensitive spectrograph. As you see, we need to use the ID for plant. Here, as you see, this is ID for Arabidopsis plant. You just put the ID here and click on the lookup, and this will give you the putative expression or cell localization, and this is related about this gene. As you see here, these genes already located and highly expressed in cytosol, mitochondria, and blastide. And the expression level starting from yellow to at minimum level to go deeping to the deep red color. This is maximum expression level. Here also, if I have the VAR database tools can help us to determine or take fast and serious decision about if I have two genes and need to select between these two genes, which gene is suitable for our experimental, or what is the expected if I try cloning and transfer these genes. As you see here, this is gene high expression in the stage of flower, and this is low expression in dry seeds. On the other hand, the other gene highly expressed in pollen grain and the later stage about embryo development and in dry seed. As you see here, if also I have so many genes belonging to the same family, like MAP family here, as you see here, this is 6 genes from Arabidopsis plant. For example, MAP4, MAP16, MAP12, MAP14, MAP24, MAP30. Okay, each gene from this have different expression profile, putative expression profile. So this is give us fast and quick decision which gene I will be select if this is gene belonging to the same family and have the same function, which gene is suitable for me. As you see here, if I study this is leaf and this is stage, okay, this is gene suitable for me at this stage. Okay. And also this is database give us good tools for study gene under different time condition, for starting from 0 hours, 24 minutes, and 15 minutes, and 1 hour, 3 hours, 4 until 24 hours. This is yellow lines represent about the day or about the light, and this is the dark or black color represent about the night or about the night. Okay, or about the dark. Okay, so here if we do comparison between these two genes, the HS1 and the HS2, under the absolute level of UV light, and the other gene under the relative level of UV light. Here we found the HS1. This is highly expressed under at 3 hours, and the expression level decrease after 6 hours. On the other hand, the DHS2, this is normal expression at all the time, at the night or the day. Okay. Here also example for plant related belonging to the Gramineae family. Here we can study this expression in all stage about plant development and all part of plant. For example, you can study the expression, putative expression of these genes in first node and first internode. Here's at different time. Here, as you see here, this is 10 day after germination, and this 17 days after germination, 27 day, and this 35 day until 16 days after germination. As you see, I don't know this is you can see or not, the highly expressed in the node and the first node we get after 35 day after germination. And also in the leaf, we also as you see here, this is gene highly expressed after 10 day from day after germination. And here in young leaves, this is highly expressed compared to the other leaf in the plant. Material and the method. How we can use this? This is depending about if our plant is non-genome or unknown genome. If our plant is non-genome, this is you can just use the ID, for example, for Arabidopsis, for tomato, for maize, for rice, for barley, for wheat. Just only use the ID and go to apply in this database. On the other side, if your genes or your data get from unknown plant genome, unknown genome plant, okay, for example, here we can search, get the ID about, for example, this gene, DXR, okay, search here, for example, DXR in Sylvia. We search about these genes, we get these genes. We just only need a little information, just only the amino acid sequencing or nucleotide sequencing about these genes, okay? We can use the one of them, okay, or can use the both of them, is okay, it's up to you, okay? So this is the gene, okay, I select about the sequence from NCBI, National Center of Biotechnology Information, and then go to the other database, phytosome database. Okay, and from the list of species, I select which species which species I need to perform. Okay, so here this is if I need to get information related with Arabidopsis database or related about glycine max, related about as you see here tomato, okay, and Josebium, cotton, or hordium, or wheat, or other brassica, or other plant. Okay. So this is already select here, and I paste my sequence here. Okay. After that, if I used, be carefully, if you use the nucleotide sequence, you just select the target type about genomic. If you select the amino acid sequence, you just select about target your type about proteomics. Okay, and hit here. This is give us high similarity, and I just all that I need from this database, just the ID. Okay, this is the gene I already selected from Sylvia officinalis. This is medicinal plant. Okay, and I just only I need the ID. Okay, I just select the ID from this database and phytosome, and then go to the bar database. Here we have two ways. Okay, the first way is about use Arabidopsis electrophorescent pictograph browser. Okay, as you will see here, you just paste our sequence ID here, and then go here. This is give us the result about where is our gene highly expressed in which tissue and which stage and which part of plant. Okay, and from data source I can select which we can select where is I need to search about my gene expression. Okay. From here, we can search about our gene expression under abiotic stress or biotic stress, about DNA damage, about embryo, about garden cell, about hormones, whatever I search, okay? And also for here, I select about abiotic stress. This is control, and this is my gene expression under putative cold. Okay, stress of cold at 4 Celsius degree, and here at 300 millimolar mannitol, okay? As you see here, my gene is starting low expression, start to go high level of expression. Here the maximum is here, I think about 1 hour. I don't know, this is already clear for you to see or not. Okay, compare with control. And I can get my data in all Arabidopsis tissue. Just click here to collect the data from table of expression value. If I need my data in chart, just click here for chart expression view here, and I can see what is the tissue and what is the expression level. Okay. The other way, I used ePlant new version from bar tools. Okay, this is can give us more information and more details about my target genes. For example, just as I said before, just I get the ID from phytosome. Okay, and then go here and paste my ID, and then search. This is will give you first the heat map viewer. This is consists from multiply about small charts, and every every small chart from this represents the highly expressed in which tissue. For example, here in this is part, this is different plant part. Here, all of this is under microscope. Tissue can't see by your eyes, just under microscope, and this is part related about the cell. And here give us gene information viewer. For example, this is ID, this is the XR gene, and be careful because some gene have different name or have other name. Okay, so which name you used? Okay, both is the same as belong to the same gene. Okay, and this is alleles, and this is full name, and this is brief description, and give us a small summary about my genes, and then give us location about genes. My gene located in chromosome number 5. And starting from this number until this number, okay, and they give us DNA sequencing, and also give us publication viewer. Publication viewer is all research or publication related about my gene or mentions our gene. This is already listed by the author name, following by years and journal and title of this is manuscript or article. And at the end, as this is PubMed and this is active link, you can get the full paper or full research by hitting this is link. Okay. After that, they give us navigator viewer about our gene. This our gene. Where is the similar gene found in other plant? For example, here our gene detected in Arabidopsis, the nearest gene found in poplar plant, and then Medicago trincotilla. This is like alfalfa, and then soybean and grape, tomato, potato, maize, rice. Okay, as you see, and this is phylogeny. Okay, and if I need to save this is expression to use in my article as supplementary data or the main figure in my article, just click here, and this is icon like camera. Okay, and you can save by two extension, PNG extension or SVG extension. Okay. After that, they give us other tissue or other or relative expression of my gene in other tissue. For example, this is tissue specific embryo development, as you see in different stage about embryo, globular, heart, torpedo stage. And here, as you see, our gene already highly expressed in root, okay, more than the cells that already will be developed to cotyledon in heart stage. For our friends that already interesting about tissue culture and secondary metabolism, as you see here, our gene, this is control, our gene highly expressed under this concentration from ABA hormone, and for other substance like DMTU, this is about 5 millimolar from this is not highly expressed. On the other side, this is highly expressed, also get highly expressed in 50 UL molar from ABA and the 5 millimolar from DMTU. Okay, and this is tissue specific stem epidermis electrophoresis spectrograph. This help us to get where is our gene expressed in stem epidermis. This is in top of a stem or bottom of a stem. This is very important. Okay. And also, this is, as you mentioned before, this is give us our gene expression, biotative expression level at abiotic stress. This is, as we mentioned before, this is starting from 0 hours until 24 hours. This is in light and this is in dark. This is okay, and this is control, and this is under abiotic, different abiotic stress. If I need to comparison about the expression stress about our gene under different condition, for example, cold, osmotic, salt, drought, oxidative, UV, wounding, and the heat. Okay, as you see here, this is map can help us to predict where is our gene were highly expressed in which time and at which abiotic stress. This is also can give us about cell electrophoresis spectrograph. Our gene already located in blastoid and the highly expressed in blastoid, and they give us localization score. Okay. And this is for our friends that were that interesting about cytology and the mutation. This is give us information about our gene located in which chromosome, as we mentioned here before. Our gene located in chromosome number 5, and which arm, long arm and short arm, and you can calculate the distance between the centromere and the location about your gene, and also give us the full size about chromosome, as you see here. Okay, and also interaction viewer, as we mentioned before. This is give us the relationship between our gene and the other gene in the same plant. As you see, this is our target gene, and have interesting in between. This is 3 genes interaction, and they give us 3-dimension molecules for our gene. Give us alpha helix and beta sheet and the loop, and they can change which form I need. Okay, and also I can change the color, and I can print this 3-dimension of our genes. This is give us other database that we can use. Okay, bar database and our scientific research. Okay, we already used the bar to predict to get some information about our gene before we do the practical research. As we mentioned here, this is our last paper that already accepted in this month, alhamdulillah, under this is accession number, and this is under the title of molecular characterization of NAD depending franzol dehydrogenase. This is Cisco terpenes synthesis from Salvia officinalis. As we predicted before we start to our in vitro work. Okay, we just predict our gene. Our gene will be in plantlet, will be help plantlet to growing fast. Okay, and our gene will highly expressed in most of tissues. Okay, and also starting highly expressed in earlier stage of food. Okay, and our gene will highly expressed in top. Okay, of stem. And our gene located in cytoplasm. Okay, then highly expressed in blastoid and the mitochondria. This is our result we get after we cloning these genes from Salvia officinalis and transfer to Arabidopsis plant. As you see here, this is right. Our gene, our plant, transgenic plant, start to growing faster compared to the control, and here highly expressed in boots. And if you see the shape or the form about the leaf here, the base leaf here in Arabidopsis, you see this is highly expressed, as you see. So this is a database can show you the way, by using this database you can predict the behavior about our genes and what is the expected from transfer the genes to the plant. Okay. The other example we already started to do in silico analysis about the other gene. His name 1.8 synol synthase. This is from Salvia ornithatica and transfer to tobacco plant. Okay, and these genes already will help the plant to growing faster compared to the control wild type. Okay, and as you see here, our gene highly expressed in all the tissue. Okay, if I see here, this is in the middle of this expression. Okay, so this is genes, and confirm this gene will for 1.8 synol synthase. This will be highly expressed in all the tissue. Okay, after we collect this plant and do terpene profile for this plant. Okay, and they start to do insect force feeding assay to test the effect about our transgenic plant on the feeding about the insect. We found our transgenic still have a little damage compared to the control after different time. Okay, this is the other paper we used. We used the bar database to collect some information that help us to predict and also in the discussion about our result. In this work, we used cooperation. This is work cooperation by Professor Dr. Ahmed Al-Quda, and in this paper we start to do genetic diversity about virus or different accession from tomato under salt stress using SSR marker. Okay, by doing some analysis we get the genes that related with SSR marker, and these genes we impressive. These genes related about different important gene. Some of these genes related about transcription factor or belong to is kind of transcription factor. Some of these genes related about membrane, and some other related about energy, and some about related about photosynthesis in plant. As you see here, we get the idea about these genes from tomato, and started to use the bar database to predict our gene expression, mutative gene expression. As you see here, this is the first gene highly expressed in all the leaf. The second gene is highly expressed in roots. Okay. The third gene highly expressed in all the upper stage of leaf. Okay. And also we get information about these genes, these 4 gene localizations. This has helped us to discuss our result, and why this is gene, or specifically molecular SSR marker related with these genes. Already we get in the plant resistant to plant tolerance to salt stress. The first gene highly expressed in cell membrane. The second gene is highly expressed in mitochondria. The third gene, this is transcription factor, highly expressed in nucleotide. The fourth gene highly expressed in plastide. Okay. Also, we get predictive information about highly expressed of these genes under different living promoter from Arabidopsis, as you see here, to determine the active site on the high expression of these genes under different root cell type, as you see here. Okay, every gene under 11 different promoters, as you see. This is highly expressed in some, and this is also can help us to discuss where is the genes, and why this localization of these genes is very important, especially in different root cell. Okay, this can help us to explain these genes and the function of these genes. Okay, this is also have information for if we have here some participate interesting about study human electrophoreceptograph or mice. As you see here, this is TNF gene. These tumor necrosis factors. Okay, we can get information about these genes under different data source. Here genes we can about respiratory, as you see. This is high expression in different tissue and low expression in other tissue. And also by change data source, we can get more information. This is data source. This is our gene highly expressed under nervous system, as you see here. Okay, and he's at the reproductive system. And the last one, skeletal immune digesting system. And this is the other genes related about catalase protein coding gene. This is can be studied in mouse. Okay, thank you.
Thank you much, Dr. Mohammed Ali, for this very interesting lecture. And now we have like 10 minutes for discussion, or maybe more. If you have more questions, we have time for these questions.
Okay.
You can write the question and I can read it for Dr. Mohammed Ali, or you can unmute your mic to ask him directly if you want. Do we have questions? No questions?
Okay.
So it should be clear.
Okay.
We can wait just 1 minute, 2 minutes for.
Okay. Okay.
So we have a question here. Please, how do we get the package?
Okay, thank you very much. Please, how do you get the package? Oh, this is not related about package. This is available website. This is free website you can use. If you go here, okay, this is the bar. I will say copy and search. Okay, as you see here. Oh, this is the link about the bar. Okay, this is free. You can use and get more information.
Okay, nice, very good.
It's clear? Yes. And also, if you go by my account in ResearchGate, you will find this is BBT available for everyone. It's free. You can download and you can use. Just you will find a little modified.
Okay, great.
Okay, thank you. For
So we have very a lot of thanks for you from the audience.
Thank you.
No more questions.
Okay.
I think Abdullah, please unmute the mic for the attendees so anyone can ask his question, and
Yes.
anyone can raise raise the hand, and Dr. Amir can manage this.
You are welcome, Ubra. Assalamu alaikum. I am asking about the different taxation number of some genetic. It is effect on the result of the same genes. Just a little bit because this is Dr. Samir. Okay. Just a little different because you see, if you see here for different. You see here. Okay. This is different taxation number. Okay. This is more clearly. This is MAP. Okay. If you search about MAP, this is each one from these genes belong to the MAP family, and this is have different taxation number. But this is all belong to the family MAP family. You understand me? We need another lecture to teach us how to use this different online tools, please. Dr. Iman Hassan. Okay, Dr. Iman, I think you can manage this with Dr. Ahmad, and it is my pleasure to give you more literature about this and other database.
Okay, we have Dr. Mohammed Samir is raising his hand. We can have his question now.
Thank you.
Okay, welcome.
I'd like to thank you for the organization and for this nice presentation, for all the organizer and Dr. Mohammed Ali as the presenter.
Thank you.
Welcome.
My question is about do you expect the expression profile of death of a gene from the species that are not included in the BAR database?
Yes.
Has the same profile as that in Arabidopsis?
Yes, this is depending about the plant and about the gene, Dr. Samir. As you see here, we talk about the other way about a known and unknown genome. Okay, for example, this is a gene already expected here. Predict, as you see, yes. Oh, this is from other organisms. This is from Sylvia officinalis. Okay, plant. We predict all this data before we start cloning and transfer these genes. You understand me?
So you can't search the bar for
I search it.
Gene. You search for the similar allele or the similar
yes.
You expect the profile, the expression profile for this gene to be similar as that of Arabidopsis.
80% from our work just to expect and to save the money and save the time. You understand me? And also this is not just only the bar. This is so different database. We used in our lab, okay, to predict and to confirm. For example, if this is, this gene related about terpenes, okay. So which kind of terpenes, mono, cis, and di, and this is the host plant. I will start to transfer my gene. This is have the same gene and have the same substrate, okay, or not. You understand me? And by the way, this is my gene. How many motif and the domain having these genes, and what is the interaction between these genes and other gene? Okay, so we predict these genes or behavior of these genes before we transfer. We get more than 80% from our computation. Okay, it's clear, Dr. Amir? Dr. Samir?
Yes. Thank you.
Welcome.
I have a question myself. Sorry.
Yes.
So how do you do the transformation? Can you give a small brief about the methods? And another question: do we, if for example, if we have the same gene in the other plant but different alleles, do you think that it will have the same effect if you transfer the gene?
Okay, good question. Okay, so Dr. Samir, yes, you asked this question.
Amir.
Dr. Amir, okay, yes.
Yeah.
For Arabidopsis, it's just easily, okay. You just have wild type seed and growing. This is the seed under the condition, good condition, okay, and they get plant. When your plant start to flowering, okay, you use you at the same time you get your gene and transfer by gateway protocol, B donor and then BBG7, and then you transfer your gene to agrobacterium tumefaciens. We used here EH 1.105, okay, and then we just deep the flower, but not the flower at this stage. You know, this is the early bud stage. You deep the flower with this is small or earlier stage of boots, and this is solution, okay, for several times, okay, and then keep this is plant in good humidity condition about 3 days in the dark. After that, you stand up your plant and start to growing, and after 1 week you repeated this is step again, okay, and after that you let your plant growing and added NPK for feed your plant, okay, and after that at the end stage about seed production, okay, after plant is dry, and this you collect your this seed, okay, and the try to transfer try to verify your this seed, and try to growing your seed again, and use pasta solution to say for this is depending about which resistant gene in your plasmid. Yes, resistant gene in your plasmid. We here use the pasta. Okay, so
Okay.
I try to spray pasta solution in this is plant. So the survival plant, this is already positive plant, have my genes, okay, or have just have the bacteria. I not confirm yet this is have my gene or not. Just this is have my plasmid, okay, and the dye plant this is not have, okay. So this is the survival or growing plant, okay. I shift this is plant to new boots, okay, and start to wait until plant is growing for maybe about 2 weeks, and then take leaves for QRT and for DNA isolation, and try to use my primer, specific primer for my target gene to detect this is positive or negative, okay. And the other way we use if you have available fund, you can direct use semi-quantitative QRT, okay. After that, you have transgenic plant, positive transgenic plant. Okay, this is the first question. It's clear?
Yes. Yep.
Okay. The second question. Okay, we do a little how to say calculation. What's this meaning? Okay, this has happened with my friend. My friend is already in old lab start to cloning one gene and transfer these genes to some kind of tobacco plant. Okay, this is so different type of tobacco plant. So he just didn't take care, just transfer these genes to plant, and he's searching about substrate many times. Okay, everything is okay, transformation is okay, the DNA as your gene already insert in this plant, QRT is okay, but the problem about metabolic, this function, he didn't found the function about his. Why? Because he didn't do a competition before he start to transfer. Why? This kind of tobacco my friend is used. Okay, this is not have the substrate or have the different substrate. For example, he working about phenol, for example. Okay, and they transfer his genes. He think he will get high phenol concentration from his tobacco plant, but the problem, okay, he didn't get phenol, so he think his work is not good. My professor tell me, Ali, search about this point and do calculation. I do calculation. I found he do mistake. He's searching about phenol, but the substrate related with his gene and this kind of tobacco related about linol. Okay.
So he has the enzyme.
Yeah. So have different substrate, different gene function. Okay. So he transfer gene related about phenol, for example. Okay. But he found the high concentration from linol, but he didn't care about linol. He just searching about phenol. So this is very important. Do this calculation before you transfer your genes. Okay, so at the final, okay, I said yes. Please give me your data. I searched in this data, rearrangement again, the raw data. I found this is the problem. Your gene, okay, in your target plant, for example, rated about phenol. But when you transfer your gene to this kind of tobacco, okay, you will get high level from linol, for example. Okay, so that is the point. It's clear?
Thanks, doctor. We have a question here from Dr. Amira. She's asking, did you try to use this database for wheat? And not sure that it contains information about biotic stress. Is it not available? If no, do you know how can I get this?
Dr. Amira, we work now in one research with some group related about the wheat, but we just didn't use, how to say, let me tell you. You see my screen now?
Yes.
Dr. Amira? Okay. Okay, so this is the only available data for wheat. You understand me? You need to get accession. Okay, if we just only, okay, here, go. Okay, but the problem for biotic or abiotic, if you need to get more information, this is you need to how to say, this is depending about the available data. For example, we get for Arabidopsis Y because this is transcriptome is already done several times, and this is many lab in around the world working about Arabidopsis. So if you need so if you need to study any genes, it easily in any lab at the world, just transfer to Arabidopsis, transfer. So get more information or predictive information. Most of these genes is not related about Arabidopsis. This is from other organisms that we already transferred to Arabidopsis. Okay, so in this you found this early, and here you see this is wheat abiotic. This is the only available. Salt, drought, heat. You understand me, Dr. Amira? You can see? Okay, and this is embryogenesis. This is the only. To cell embryo, embryo, okay, and this is the available data for wheat. And you can see his expression. This is, yes. This is the only. If you need to study these genes from wheat under how to say, under cold or under salt, UV, other, you can use the Arabidopsis database. Okay, this is a little complicated. We're working in some genomic of some plant genomics, and they make comparison genomic between this plant with Arabidopsis. Okay, to found the similarity genes, because Arabidopsis have so large available data, can help us to explain why these genes or function, to study function of these genes. It's clear, Dr. Amira?
Yeah, I think so.
Okay. Yes, I do. Yeah, okay, Dr. Amira Murad. Okay.
Dr. Lamia, thank you for this good lecture. My question is, if it's possible to extract the genetic material from the plant, from any plant part other than the growing tips, and if it is necessary to be fresh part, if we could use dry plants, especially seeds, for extracting the genetic material.
Any plant part other than growing tips, if it is necessary to be fresh part. I think in old lab, some friend, our neighbor lab, working in study how to say, brassica seeds. Okay, so not just very dry, just started to extract the embryo from the seed. Okay, and then starting to collect or isolation protein. Okay, and messenger and RNA and DNA. Okay, from different stage, from seedling. You understand me? Just dry seed, we need to know the content of these seeds. Okay, the other working about the boot and the stage and the development of seeds in the boot and brassica plant. Okay, so this is work is so a little hard. No one can do this. You understand me? especially dry seed, because to crush the seed and get extract the embryo, because you will found how to say, you will found your genomic material or genetic material in the how to say, in the embryo, just only because this is have copy from this is genetic information data. Okay, and also depending about the seed. Okay, this is oil seed different from the other seed. Some seed have high how to say high amount of oil. Okay, other seed have high amount of starch. You understand me? So this is depending about the seed and what you need to study. Okay, Dr. Lamia, it's clear?
Yeah, clear. Thank you very much, Dr. Mohammed.
Welcome.
I think we are done with the questions now. I would like to ask all the attendees if they want like certificates, they have to register on the on the link in the chat room here, if you want. And thank you very much again.
Thank you, Dr. Amir, and thank you, Dr. Talat. Abdullah Talat.
Thank you, and goodbye.
Thank you. Goodbye. Goodbye.
Thank you, Dr. Mohammed.
Welcome.